Good afternoon. Thank you for the invitation. I have a little bit hard time because you're after lunch. It's the worst time to talk with you. So, and I'm going to talk about molecular, but not only about molecular stuff. So, what are the questions that we are interested in? The question that we are trying to answer is how taste internal representation is encoded, associated, consolidated, and retrieved in the mammalian cortex. And of course, this is a very vast question. And the second one is, can we translate our better understanding for the question above to fight mild cognitive impairment and dementia? So what I'm going to talk with you about is, since I had also lunch, I prepare a surprise in the middle of the talk, so we will all wake up. And what I'm going to talk with you about, first I'm going to introduce you to the taste system, which we like a lot. And then I'm going to tell you about a new work that we're trying to answer how the internal representation of a given taste is waiting in the brain for an association. Surprise. And then I'll talk about from memory consolidation and translation regulation in the cortex to involvement of a specific protein, eukaryotic initiation factor 2 alpha pathway in aging and Alzheimer's disease. I'll give a little bit background from our publication, but mainly we'll give you unpublished results. So why someone likes taste system? There are multiple reasons. I'm coming from behavioral perspective, and I'm trying to understand molecular and cellular mechanism of learning and memory. So taste learning is highly natural behavior. It's robust. If we sample novel taste to a certain amount, we remember it for a very long time. It's one trial, so I have a T that I can measure from and at. It involves incidental learning, and I assume most of our memories are incidental kind of memories. It is cortical-dependent, and we heard today long-term memories are supposed to be, or at least in part, stored in the cortex. It's one may say absolute definition, milliliter and molars. However, still, even for a novel taste, you have the temperature and the texture of any given taste that we are trying. It's easy to measure. It has a good electrophysiological model, which I'm not going to talk about. But the main reason that I'm working on a taste is taste as a sensory information is coming always with a valence, and as such is different from other sensory modalities. And I think that a good question is, when do we have a memory and when we do not have a memory? Then when a given sensory information is coming with a valence, then the chance that we will have long-term memory is much higher. And we try to understand this coincidence between the sensory information and its valence. And the associative learning paradigm, which is called conditioned taste aversion, is a weird kind of association that I think can teach us about regular association. How do you measure taste in human is really interesting, but today I'm going to talk about rodents. So one way to go is just preference. You give a mice or rats novel taste, and then a few days later you ask the rat or the mice what does it prefer. And with time, if the taste did nothing wrong, it will consume more and more from the given taste. And if we talk about better memory, then you can calculate a preference index, which is the novel taste divided by total consumption. The negative form of learning is association between novel taste and malaise-inducing agent, for example, injection of lithium chloride to the belly. And following this association, you can wait days, weeks, or more. And then again, you ask the rat, what does it prefer? And it will avoid the novel taste that was associated with the malaise-inducing agent. Now, as I said before, this is weird kind of association because usually when we talk about association, time scale is seconds. Many seconds is a long time for association. Here, for highly reasonable evolution perspective reason, the time is very long. But from our model, it makes a little bit of a problem because it can be many hours between the novel taste, between the conditioned stimulus and the unconditioned stimulus. And there is another way to measure taste, because of course we experience both positive and negative kind of taste learning. If before the negative learning we learn that the taste is safe, we will be less aversive than if we had only the conditioned taste aversion, and this is called latent inhibition of conditioned taste aversion. And you can calculate the aversion index by the familiar taste divided by total consumption. And with CTA, the higher the aversion index, the better the learning. With latent inhibition, it's the other way around. And this is a good reason why to work with in rodents in taste learning. You can see here the aversion index. These are n equal 4 rats that were associated. This is CTA. These are animals that were injected with saline. So this is like second exposure to saccharin. And this is animals that were pre-exposed before. They are aversive compared to control, but less aversive than animals that had conditioned taste aversion. Okay, so neuroanatomy. I'll speak mainly about the cortex. When we talk about taste, taste is coming from the tongue via these 3 cranial nerves to the brainstem, pons, thalamus, and the gustatory cortex, which reside within the insular cortex. You can see it also here. If we try to understand better the insular cortex, there are few models about how the gustatory cortex is working. And one of them is that there is some kind of somatic map or taste map in the cortex. There are indications for here and to there. And there is another possibility, is that the valence of the taste is changing the topography of the representation of a given taste, and it can shift with our experience. It's important to know that we need the gustatory cortex in the insular cortex both for learning taste, but also for retrieving the memory of whatever we have learned before. If we zoom a little bit more into the cortex, the gustatory cortex is agranular cortex, which means that you have disappearing of layer 4. You can see here inflated cortex. This is the piriform cortex, and you see the transformation from granular, insular cortex, this granular disappearing of layer 4, and the agranular cortex. Of course, the cortex is connected between itself, the different agranular, dysgranular, and granular, but also with other brain regions. And we are looking specifically at prefrontal cortex and basolateral amygdala. So this is for the neuroanatomy. What we're trying to do from molecular perspective, we try to tell a story of correlation and causalities by the following. If time 0 is when the animal experienced the novel taste, we try to look for correlations following this experience, and we can see release of neurotransmitters. For example, Mona was talking about acetylcholine. You have a prolonged, like 30, 40, 50 minutes release of acetylcholine in the gustatory cortex. And then you have different post-translation modification phosphorylation events that you can see here. And also you have protein expression, as you can see here. This is a review from 2011. We are interested also in transcription, both coding and non-coding RNA, as Mona described them just before, degradation regulation, translation regulation, and post-translation regulation. So today I'm going to talk with you mainly about translation regulation. Now, of course, most of you, I don't need to remind you the gap between molecular, circuit, and cellular mechanisms. And of course, we're trying using in vivo calcium imaging, for example, in collaboration with Andreas Luthi, who is sitting here in the audience, and also electrophysiology to try to bridge the gap between the molecular and the circuit to cellular mechanisms. One major issue that we have is that the taste and insular cortex are not following one of the rules that other learning paradigms are following. And this is that you don't see expression of immediate early genes in the cortex following learning. So you can see here a Venn diagram of RNA. We looked for RNA, and we looked also for microRNA expression in the gustatory cortex following taste learning. So the groups are, W is a familiar taste, 1 hour, or familiar taste, 3 hours. N is novel, 1 hour. versus novel taste 3 hours. And as you can see here, this is novel versus familiar, novel versus familiar. And what immediately you can see here, that there is a clear change in mRNA expression just because of the drinking, the kinetic, the time following drinking, but not the novelty effect. I mean, there are very few genes, like 1 hour is only 10 genes that are changing following the novelty component of the learning. It's even more complex in the sense that if we compare to water-restricted animal, and that's what one should do, you can see that just because of the drinking, you have a high expression of ARC. And ARC is a good indicator for activity in principal cells, in excitatory cells. And if you look on the novel taste, actually what you see a little bit of a reduction from the high induction of ARC. What I want to say, I'm not going to talk about this, but I want to say that many people that are working on incentive drinking, I would be very careful with it, because I think that either the anticipation or the drinking itself change mRNA expression not only in the gustatory cortex, but also in other brain area. However, with post-translation modification, for example, ERK activation, one can look at connectivity. For example, if we look at ERK, and phosphorylation of ERK is its activation, and it's highly important in cancer research, but also it's necessary for formation of long-term memories. So if you inject ERK inhibitor to a specific brain area, you will inhibit the formation of long-term memories. If we look for the correlation, you can see here, this is the amount of ERK activation. You can see here that following novel taste, we have more ERK activation compared to familiar same saccharin or water. And we can do just immunohistochemistry to identify the cells that have more phospho-ERK. As you can see here in the cortex, these are principal cells. And this group is the novel taste. This is water. This is familiar saccharin. So animal drank the saccharin for more than 5 times. And you can look at the quantification of the data. And if you look just over here, this is left versus right. But if you look at total, you can see that most of the changes in the phosphorylation of the ERK, you can see it in the agranular insular cortex, but not in the dis or in the granular insular cortex. And you can combine it with retrograde tracers. And as we said before, we care about the connection from medial prefrontal cortex, from basolateral amygdala, and from the thalamus. And you can look at the insular cortex for the cells that are sense projection, axons, to this different brain area. And you can ask which cells that send projection to this different areas also have elevation in phosphorylation of ERK. And as you can see here from the data, when animal experience novel taste, there is clear increase in cells that sense projections to the medial prefrontal cortex, in comparison to animals that experience taste that is negative. So this animal had conditioned taste aversion. Now the taste is negative, and they are drinking it. So when you look at here on the taste that is negative, you can see that the cells that send projection to basolateral amygdala express much more phospho-ERK. Okay. So let's go for the first question. And the first question is a very basic one. We know that there is conditioned stimulus, novel taste, unconditioned stimulus. It takes very long time. So the question is how the memory trace is waiting for the unconditioned stimulus for such a long time. And we are trying to follow the pillar in the field. And let's read this one. A connection is formed in the nerve system not between conditioned stimulus and unconditioned stimulus, but between the sensory after effects, i.e., trace of the conditioned stimulus and the unconditioned stimulus. Okay, so we try to identify the ghost of the conditioned stimulus and try to see whether it stays there for such a long time. And another great scientist that, of course, you know have is if you can look at the synapses, changes at the synapses, they may explain the change that underlying the mechanism of the conditioned stimulus waiting for the unconditioned stimulus. Okay, So in order to answer the question how novel taste memory trace minutes to hours following perception is maintained for an association to occur with the visceral information, we need to do the following. We need to identify a synaptic, molecular, and/or cellular process in the relevant brain structure with the correct time frame. The process should be both correlative and necessary. And if possible, the process should subserve the memory of the taste trace for an association and not taste memory per se. Okay, not just the memory of the taste, but for the association. And this is a project of a PhD student, China, in the lab. And first of all, we looked at the behavior to understand better the behavior, the time frame of the behavior. And what you see here, China did a conditioned taste aversion in different time, 1 hour, 2 hours, different ITIs before the injection of the lithium chloride. And this is the result that you obtain. What you obtain is that you can see clearly that there is 3 hours time frame and longer, 4 to 8 hour time train. And when we did a statistical analysis, there are clear differences between 1 to 3 and 4 to 8 hours time trace between the short and what we call the long-term trace of taste. This is true also for the extinction. So in extinction, you give the animal again and again the novel taste, which is not novel anymore, and they will consume more and more following the conditioned taste aversion. And you see also here that statistically, the two groups behave differently. So the 1 to 3 hours, this is the 1 to 3 hours, and this is the 4 to 8 hours. So we're looking at different possible mechanisms, and the idea is we're looking for some kind of memory in the system which can maintain itself. And it's known from molecular perspective in other cells that kinases, you can have positive feedback loop with kinases, either few kinases, A, B, C, within themselves, or some kinases can do it to themselves. For example, the calcium calmodulin-dependent protein kinase. So you see here the postsynaptic density. You see the CAM kinase 2, which is a major protein in the postsynaptic density. And following its activation, because of calcium influx from the NMDA receptor, CAM kinase can phosphorylate itself, be active, and then following its activation, it can either phosphorylate AMPA receptor, or it can insert more AMPA receptor to the synapses, to the synapse. Now, following its activation because of calcium calmodulin, there is its autophosphorylation site on threonine 286, and this kinase, its kinases, is going to stay active for a long time and retain the memory of the influx of calcium in the synapse. In addition, there is an inhibitory peptide that produced by the cells themselves to inhibit this process, and we can use part of it in order to inhibit the calcium calmodulin-dependent protein kinase. Of course, there is a lot of data regarding the role of calcium calmodulin in different synaptic function and behavior. So what first China did, he did synaptosomal fractions and look at the expression of the different proteins, including the phosphorylation on the 286 threonine site, but also on inhibitory site, which reside on the threonine 305. And this is the time dependency that he obtained. If you look here, you see here that if you measure threonine 286 phosphorylation, and this is the time following sampling novel taste, you see that there is an increase that stays for many hours, but go back to basal level 5 hours following sampling this novel taste. We don't see any difference on the inhibitory site of CAM kinase, and we don't see any difference on the expression of the CAM kinase itself. So this is the synaptosomal fraction. So you have a clear prolonged phosphorylation of the CAM kinase at the synaptosomal fraction in neurons in the gustatory cortex. It is NMDA dependent. When you block the NMDA, you don't have this increase in phosphorylation. Now, we want to inhibit, to show causality, we want to inhibit this phosphorylation of CAM kinase. And as I said before, what we can do, we can use this peptide, inject it to the insular cortex, and look for the behavioral change. And that's exactly what China did. So if this is the taste, conditioned stimulus and unconditioned stimulus. If you inject the CAM kinase inhibitor just after experiencing the test, then you have a clear reduction in the conditioned taste aversion. If you now increase the time between the conditioned and unconditioned stimulus, you still have a clear effect on the conditioned taste aversion. However, if you inject the inhibitor 4 hours following the experience of the novel taste, you don't have any more effect, which means that you need the CAM kinase within the time frame of the short, you can call it short, the 1 to 3 hours time frame of the taste learning. So as anticipated, we were looking also at the expression of glutamate receptor in synaptosomal fraction or phosphorylation, and we found that there is no increase in phosphorylation, but there is a clear increase in GLUR expression in the gustatory cortex. And if we look for correlation between the phosphorylation of the CAM kinase and the expression of the GLUR1, we see that there is a clear correlation following experiencing of the novel taste, but not a familiar taste. And we want to know whether this insertion of the GLUR1 into the synapse is CAM kinase dependent. We inject, again, the CAM kinase inhibitor to the cortex, and you see that the increase in GLUR in the synaptosomal fraction that you can see here, you don't see it anymore when you inhibit the CAM kinase in the gustatory cortex. Okay, so now we actually asked, do you really need the AMPA receptor just by pharmacology, and do the same manipulation, similar time frame, just injecting CNQX to the gustatory cortex, and indeed the results were just the same. You need the GLUR1 for the short-term association, but not for the long-term association. Okay, so to summarize this data, what we think is the following. Conditioned taste aversion is 2 different kinds of learning. I don't think it's only conditioned taste aversion. I think this is true for other learning paradigms as well. So the first one is cortex-dependent, and it's robust, but it lasts short. The other one is longer, but it lasts, it's weak, but it lasts longer. We heard before a beautiful talk about the role of the hippocampus. So, of course, other parameters can affect any learning paradigm, including conditioned taste aversion, which used to be considered hippocampal dependent independent. So when we analyzed, it was published a year ago, the Tonegawa's NR1, so non-functional NMDA receptor in dentate gyrus CE1 or CE3, we can see effect of how long ago the taste was eaten, where, and so on. So you have this additional effect of the strength of the memory. Now the question is, okay, so we have a mechanism. Of course, we need to identify the cells, but we have a molecular mechanism for maintaining the memory of the taste, waiting for the association. Is it exclusive for the taste memory waiting for association, or also for the taste memory itself? And the answer, it's exclusive for the taste trace waiting for the association. Because when we inject either APV, NMDA blocker, or TAT, or CNQX, and we measure novel taste, we don't see any effect. So to summarize this part, the taste-neurosia association that occurs with an interval of up to 3 hours differ behaviorally, both quantitatively and qualitatively, from 4 to 8 hours association time. This result suggests that there are 2 memory traces, at least 2 memory traces, one that is robust but lasts short, and the next one which is weak but lasts longer. NMDA receptor-dependent CAM kinase 2 activation, which induce synaptic GLUR1 expression in the insular cortex after novel taste consumption, is involved in generating the short memory trace, which lasts up to 3 hours for the association with the unconditioned stimulus. Now we need to move to the consolidation phase. This was how taste memory is maintained for the association. Now I'm going to tell you about the consolidation. And I promise surprise. Okay, so I hope you'll wake up at least now. And for the surprise, it's not NBA, but I put it for you. Just, of course, you know that there were NBA games not a long time ago. But the important thing, like many times in science, is what you don't see here. You don't see here Maccabi Tel Aviv. So this Thursday, there's going to be a game of the final between the best Israeli teams, and you see the blue one from Eilat and the red from Jerusalem. And though I'm in the north now, in Zichron Yaakov, I was born here in Jerusalem, and there are things you cannot change. Okay, so this was surprise, and now let's go back to the consolidation. We heard before a very nice introduction for the consolidation. When we say consolidation, we mean different things in the same word. The dogma, which has a lot of problem, we can discuss it if you want, is talking about short-term memory, and then some kind which is like molecular consolidation, and then the other consolidation, which is the dialogue between the hippocampus and the cortex, which was presented so nicely before. When I say consolidation, I mean molecular consolidation, and this is this trace. And this trace can be defined biochemically because it's sensitive to protein synthesis inhibitor. This is not a good definition, this molecular mechanism. It's not a good definition because it's a negative definition. So we and other were looking on the translation machinery in the last, I think, 12 years now to see whether it's changed following learning and contribute for molecular consolidation. And many people did their career here. So you can see the mRNA. This is the coding region of the mRNA. This is the 5' untranslated region, and this is the 3' untranslated region. And you can regulate translation in different way. When I was talking before a lot about microRNA, and you can see here the microRNA binding on mRNA, and you can, of course, regulate translation this way. In our lab, we focus on the initiation factor to alpha and the elongation factor to alpha. We don't have time to talk about the elongation. I will talk about the initiation. So when you talk about protein synthesis phases for the non-molecular, I'm not going to go into the details here. You can divide to initiation, elongation, and termination. But I need to say a few more things about initiation. So in order to have a competent ribosome, which you see here, which means you have the two subunits, the small one and the big one, the RNA and the relevant initiation factors, you need to have initiation factor 2 in a GTP form. So you see that it's transformed here from GDP to GTP, and this begins the whole process of what is so-called the ternary complex. You can see here the tRNA, the small ribosomal unit, and the initiation factor, which builds up the pre-initiation complex, and then when you have all together, the big subunit of the ribosome is joined to begin translation of a given mRNA, and then you have a release of this initiation factor to alpha, which is the key for the whole process. Also because its amount is not very high in the cell. Now you can regulate it and cell regulate this process by phosphorylation at serine 51. So if this protein is phosphorylated, you stop translation. And if it dephosphorylated by specific phosphatase, then you have more translation. And this is a key factor. Now, in the last few years, you can see the different papers that were published in this topic. And let's begin with this major paper, and I will summarize this just with one slide. So the idea is the following. As I said before, initiation factor to alpha can be in dephosphorylated or phosphorylated state. And you can regulate translation. You can have more or less protein, or even more so, specific set of protein that can help the building of long-term memory. So if the initiation factor 2 is in phosphorylated state, you have less general translation. You have more translation of specific mRNAs that have multiple initiation site, like ATF4. And generally speaking, it's more difficult to get synaptic plasticity, LTP, and LTD, and memory. On the other hand, if it is in dephosphorylated state, you have more translation, less ATF4, which is a negative transcription factor, and it's easier to have long-term memory. The initiation factor 2 is regulated, this is a phosphorylation site, of course, by different kinases. HRI is not expressed in the brain or expressed in a very small amount, so we don't care about it, but it does, but GCN2, PKR, and PERK are expressed in the brain. So let's just talk about every cell. Every cell would like to stop translation when it is in metabolic stress. For example, if you don't have enough amino acid, or if you have double-strand RNA, meaning viruses, or if you have a lot of misfolding and you have ER stress, you will activate the pathway and stop translation. This is highly energy-consuming process, and you don't want to waste it. Now, remember that what neurons are doing many times, they hijacked major pathway, and neurotransmitters and pattern of activity can activate or deactivate this kind of pathways. So now there is a very simple hypothesis. If we will reduce the amount of activity of the kinases, there will be less phosphorylation, and it will be easier to form memory. Okay, what do we need to do in order to do this? We need to identify the kinase which determine the initiation factor to alpha phosphorylation in the brain. And then we need to have tools to inhibit it or its activity. And then we need to measure behavioral, cellular, and molecular phenotypes, and we assume we'll have a better memory. Of course, we can test it also in aged animal and in Alzheimer's disease, which is going to be the end of the talk. Okay, so we made different viruses with shRNA to reduce the amount of the different kinases. Remember, the GCN2, the PERK, and the PKR, and we tested them in cells. And as you can see, we found a clone that can reduce their expression in a clear way. And when we test for the level of phosphoinitiation, we see that PERK, either in 3T3 cells or in primary culture, is the major kinase to determine phosphorylation of initiation factor to alpha on the basal level. Okay, now what we need to do, either inhibit PERK with a small molecule or inhibit it with a virus and test for learning. And that's exactly what we did. And as you can see here, this is a preference test, just as I explained before. And if animals are injected when they learn the novel taste with the inhibitor of the PERK, you have better memory. This is for the negative learning. This is the scheme. Again, you have better memory. And if you inject the virus that reduces the expression of PERK before learning in the insular cortex, as you can see here in the coronal section, you get very similar result. You get enhanced positive taste learning, and you get much better CTA. So, by the way, we're doing here weak CTA because we know that we are looking for better memory, and if the learning was 90%, there will be a ceiling effect, and we won't be able to see better memory. But it's not only that the animals have better memory. The animal actually have better plasticity because you see that they are even better in the extinction phase. You see the reduction here. So the test is also an experience, and animal learn that the taste did nothing, and actually they are less aversive on the second day. As you can see here, the shRNA reduced the expression of PERK in the brain and reduced the phosphorylation of the initiation factor to alpha. Now we can ask, do you need it for the learning or do you need it for the retrieval? And in order to ask whether you need it for the retrieval, you can first do the learning, then inject the virus, and then test the animal. And as you can see here, you don't need the PERK for the retrieval. Okay, so we can say that PERK is the main kinase to determine basal level of phosphoinitiation factor to alpha in neurons. Local inhibition of PERK in the insular cortex using a small molecule reduces phosphorylation of initiation factor to alpha level and enhances both positive and negative forms of taste learning. PERK knockdown in the insular cortex decreases phosphoinitiation, enhances positive and negative forms of taste memory, and increased behavioral plasticity. And PERK knockdown enhances memory and at the consolidation phase, but does not affect retrieval. Okay, now we can ask ourselves, does the same manipulation will work in the hippocampus, and will it also enhance memory of aged animal? Now, when you talk about rats and mice, aged animal, if a mice is 1 year, 1 year is about my time, when clearly you have decline in your cognitive abilities, but old is something like 1 year and a half. So we can, as you can see here, inject specifically to CA1 or specifically to the dentate gyrus and look for the behavioral phenotype. And I'll show you just the CA1 results in aged animal. So let's look here. This is field conditioning because we're looking for a behavioral phenotype that it has clear age dependency. So you have here the blue, red, green groups, which is 7 months, 10 months, and 12 months animals. The green one are injected with a control. The purple are injected with the shRNA for PERC. And this is the amount of freezing, all the y-axis, following trace field conditioning. Trace field conditioning means you make association between the tone and the shock, but you do it in specific trace, 20 seconds following the tone. So the animal should freeze specifically at this time. And what you can see here, you can see that it doesn't matter how you measure it. Animals that were injected with shRNA to PERC, the aged animal, behave either better than the young or like the young. You see here green are the 12 months old. You see how better they are compared to the animal injected with the shRNA to PERC. Okay, so we talked about the PERC, and I want to shift to Alzheimer's disease, but the idea is the same idea. You can inhibit one of the kinases, the PERC, or you can inhibit the PKR, and now the hero of the story is the PKR, which has to do with specifically with Alzheimer's disease. We show that when you inhibit the PKR, you have better memory. This is just a summary from the paper that was published 2013, and this is just an example. When you inject rats or mice, it doesn't matter. You have better condition taste aversion. A very important control is to inject it to mice, knockout mice for PKR. You don't see any effect on mice that are knockout for PKR, which means that the PKR inhibitor is specific. Okay, so Alzheimer's disease, which is the last part of the talk. Alzheimer's disease is a neurodegenerative disease, most common form of dementia, and it's above 95% is sporadic, which means there are non genes or clear genes to be associated with the disease. And when we began to look at this, when we try to translate our finding, you see that we can enhance memory to see whether we can also rescue phenotype of Alzheimer's disease. We found that actually the animal model of familial Alzheimer's disease, we find a lot of problem with them, generally speaking, and also we wanted to try to see what can we do to the sporadic, which is the major form of Alzheimer's disease. Now, the main risk factor for Alzheimer's disease is age. In addition, if we express in both our allele ApoE4 gene, then this is the most important risk factor for sporadic Alzheimer's disease. Now, you have a mice model of ApoE4, which means this mice does not express the murine ApoE, but it does express either the ApoE3, E3, so-called good, and the ApoE4, so-called risk factor for Alzheimer's disease. Now, metabolism of ApoE is highly complex. We can talk about it, but again, the main idea is that patients with 2 allele of E4 have up to 20 times risk to develop Alzheimer's disease. Okay, so what we were thinking? We were thinking the following. If we look at the initiation factor 2 alpha, all cells, when they have metabolic stress, phosphorylate the initiation factor 2 alpha is a read of metabolic stress readout. So all cells are phosphorylating initiation factor 2 alpha and stop general translation, and you have these specific genes that are doing the other way around. By the way, 2 of them are highly relevant for Alzheimer's disease. Now, the idea is that if age is the major risk factor for Alzheimer's disease, then we can say that age is a prolonged metabolic risk factor. So if you, for a very long time, you have this protective effect of phosphorylation of initiation factor 2 alpha, in the end, what the outcome of this might be the development of Alzheimer's disease, maybe via this mechanism, for example, high expression of base 1. But in addition, clearly it will affect your cognitive skills as we show in normal animal models. In addition, you do know that in Alzheimer's disease, the correlation between pathology and cognitive function is not that clear. This is to put it on the mild way. So, recent papers that were published say that phosphorylation of PKR, which means activation of PKR, is the best correlative molecular, it's the best molecular correlative with cognitive dysfunction. So we have a very good reason to look at PKR. And when we began the experiments, the idea was very simple. We looked at ApoE4 as a risk factor, metabolic risk factor, which means it should activate or phosphorylate initiation factor 2 alpha, and age should do the same. So we tested, first of all, aged animal. It doesn't matter which rat or mice we tested, but always aged animals have higher phosphorylation of initiation factor 2 alpha. Okay, so this is regarding age. Now, what about ApoE and ApoE3 and ApoE4? Mice that express ApoE3 shows the normal elevation in phosphorylation of initiation factor 2 alpha. However, ApoE4 don't show it. Now, we are talking about what is so-called young mice, 4 months old. So, if we don't, so these are young compared to old. So with ApoE4, we don't see it. So there are two possibilities. And one of them is that the ApoE4 has an elevation of phosphorylation of initiation factor 2 alpha already in young age. So they behave like old already in young age. And this is exactly what you see in this Western blot analysis. It doesn't matter if you look at the cortex or in the hippocampus. So young ApoE4 has an elevation of phosphorylation of initiation factor 2 alpha. Now, all the people that work on ApoE4 until this paper actually examine behavior of old mice. But we said, okay, if it has more phosphorylation, from our experience, it should be defected in learning and memory paradigm. And this is exactly the case. So you see here contextual fear conditioning. So you need to, the mice should associate between a given context and electric shock. You see here the learning curve, which looks very similar between ApoE3 and ApoE4. You see here different protocol, weak protocol or strong protocol. It doesn't matter. Young ApoE4 show less good memory, and the amount of freezing is the amount of memory in fear conditioning. So ApoE4 show less good memory if you test it 24 hours later or if you test it 1 week later. Now, we looked at the expression of all mRNA that are connected to initiation factor 2 alpha pathway, and we saw that the ApoE4 express specifically more ATF4 gene. Now, there is a subject that we didn't talk about, and the subject is, do you see in different brain regions and different cells different regulation of translation? And the answer is yes, which makes life very difficult. Okay, but here you see immunohistochemistry for ApoE4 and non-radioactive immunohistochemistry, and you see that the difference between ApoE4 and ApoE3 young mice is mainly in C1 region of the hippocampus, the region that is associated with the defect in the context, in the contextual fear conditioning. Okay, so we wanted to know whether the increased ATF4 expression is only between the E3 and the E4 mice, or maybe the E3 they have less. The answer is not. This is just in comparison to wild type mice. we wanted to know whether ATF4 expression is behaved like phosphorylation of initiation factor to alpha, namely already from young age, the animals, the ApoE4 mice, they have more phosphorylation of initiation factor to alpha and more ATF4 expression. And then we said, okay, so this is all nice, but this is mice model. We have a clear prediction, and this is that human that express ApoE4 will have increased expression of ATF4, and this is from frontal gyrus, and indeed, this is the case. So you see increased expression of ApoE4, of ATF4, sorry, in humans that express the ApoE4. Okay, so now we want to reverse the phenotype. If the phenotype is PKR dependent, we want to see, can we rescue the behavioral and the molecular phenotypes that we see, that we identified in the ApoE4 mice. And the idea is to inject the PKR inhibitor just before the learning, and that's exactly what we did. And as you remember, freezing means memory, and this is the learning curve, which looks very similar for E4 animals injected with the PKR inhibitor, E4 animal injected with vehicle, and E3 animals injected with vehicle. And this is what you see for the memory. This is a repetition of the reduction in the C1 dependent memory in the E4 versus the E3. And these are animals that were injected just once with the PKR inhibitor, and they go back to E3, or what one would call a basal level. Does it work also for the ATF4? The question is partially so. So this is the difference in the expression of ATF4 between E4 and E3, and you can see that this one injection of PKR inhibitor reduced the expression of ATF4, but did not bring it back to the basal level. Yeah, okay. So to conclude this part, pharmacological or genetic inhibition of PERK or PKR enhances memory in young and aged rodents. Chronic phosphorylation of initiation factor to alpha upregulates ATF4 mRNA brain expression in ApoE4 mice. Pharmacological inhibition of PKR partially reduces ATF4 mRNA levels and reduces cognitive impairment. And we were happy, though this is reasonably not too old data, that some drug company already replicate our results. And at the moment, of course, what a new company that is based on our finding, but I understand that other companies are doing it as well, in a way compete with this company which is based on our finding. The idea is to try to identify better PKR inhibitors in order to use them in human studies. Okay, so now we zoomed into PKR and PERK, but I want to share with you some of the questions, problems that we have, and zoom out will take also the more physiologist people here, I hope. So a major question at the moment is, what is the connection between excitatory inhibitory balance, translation regulation, and different brain function disease? What do I mean? A lot of brain diseases, developmental, but also aging, dementia kind of deficit, are being explained in the field by problems in excitatory inhibitory balance, and in other papers by defective in translation regulation. We think they are connected. I began to talk about it when I was trying to imply that different brain regions regulate translation differently. In a way, it's weird. That's what I'm trying to say. There is different regulation of translation in different brain regions, and also different neurons. So inhibitory interneurons will regulate translation differently. And from a data that I didn't show you here, but for example, when we take elongation factor to knockout mice, okay, it's elongation factor, it's another story. The phenotype that we see there, a clear phenotype, is tonic inhibition in the dentate gyrus. So it's highly, highly specific for specific cell type and also a brain region. Okay, what I want to say, in the field, it's like two different aspects of brain function and disease. And this is going to the second question, identity of the differential regulation of protein synthesis in different brain region and neuronal types. Okay. Now, another major question is, what is upstream to translation regulation? I said it before that neuronal pattern of activity and neurotransmitters, mainly neuromodulators, can regulate translation, also fast neurotransmission, of course. So we need to understand better how neurons control translation regulation, and we know very little about it. And we also don't know what is downstream from translation regulation. So only in the last 2 years, we can do kind of ribosomal profiling. We have the technology to ask not what's going on with whole protein, but what are mRNAs that are being translated in specific cells, specific neurons, following learning or other manipulations. And the last question, which is maybe too big, but I think we need to put it on the desk, is what is the interaction of protein synthesis regulation with other major pathway to control gene expression? So when I sit in another kind of sessions, more, let's say, molecular, people will talk about epigenetics and protein degradation, and will explain the same phenomena. Well, as you do know, and I'm sure you know, the cells respond to any change. And I strongly believe that when you manipulate either with mice, knockout, or whatever modification you're doing, but also when you have a disease state, when you manipulate the translation machinery, the cell respond. And it can respond in an epigenetic way or in regulation of degradation, which is another big aspect. So it's not good for papers, because if you open everything, you cannot answer specific questions. On the other hand, I'm sure that there is a clear interaction between this major regulation of gene expression. So I would like to thank the funding agencies and mainly the students that did the work. China, I talked about, and Iliana, a postdoc in the lab, Hadil, and Ifat. Here is Ifat, did most of the work that I presented here. Thank you.